Okay, as you watch this video, make sure that you have your Pear Deck open for the asynchronous kids. If you are working on this in class, you can always come back and watch this video to review anything that you might have missed in completing your Pear Deck so that when you turn it in and I grade it for completion, you actually get all your points. Our do now in class is in passive transport, molecules diffuse from high to low concentrations. In active transport, they are moved from blank to blank concentrations. Select your answer. And the learning objective is that y'all will be able to describe the process of osmosis and describe its importance to cellular integrity. So we're actually only going to focus on osmosis today, and that's going to be the movement of water or diffusion of water. And I mentioned it in the last class, but it is important that we focus on just osmosis because, well, water is important to life, and we're going to see why. So here are some words you should know. So the solute is going to be the substance that is being dissolved. So in the case of hot cocoa, the solute is going to be that cocoa powder. The solvent is the substance that the solute is getting dissolved in, or it's the one that does the dissolving. So in this case, the solvent could either be water or milk, depends on how luxurious you're trying to be. And then the solution is the final mixture of the solute that is dissolved in the solvent. So when everything is mixed together and you actually got your hot cocoa, that is a solution. So when you mix two things together and at least one of them is a liquid, then you have a solution. You can get a solution from mixing two liquids together too. So what is osmosis? Osmosis is the diffusion of water. And it's when water moves from a high concentration to a low concentration of water, which means it's going to be basically moving where it's a low solute concentration to a high solute concentration. So basically water is going to go wherever it's saltier or go to wherever it's sweeter. Okay, because that concentration is the opposite of the water concentration. And we already talked about saltiness and sweetness. And so you can think of it that way, or you can think of it based on water concentration. You got to pick one though, and you got to stick with it. And osmosis again is diffusion, and it's from high to low, which means it doesn't take energy, right? So this is what I just explained. It means that water moves from a low solute to a high solute concentration. So just the easiest way I think to think of it is wherever it's salty, water is going to go to it. Wherever it's really, really sugary, water is going to go to it to try to balance it out and even out the saltiness of the sugar in it. So for a quick note, there must be a semi-permeable or selectively permeable membrane in order for osmosis to happen. If there's no membrane, osmosis doesn't happen. Okay, then it's just diffusion. If there is a membrane, then osmosis can happen. So circle your prediction. In osmosis, you're going to think about where is there more solute, which one's concentrated, left or right? Which one has more water, left or right? And so therefore, which way is the water going to move? Is it going to move left or right? Okay. The labeling on the picture already explains that there's more solute on the right side, as you can see right here. And the higher concentration of water is going to be the opposite of that. So if there's less solute, there's more water, right? Because they take around the same amount of space. So if there's less solute, then there's more water. Now, remember, water is going to move to where it's saltier or where it's more sugary. So make a prediction with your drawing tool and draw an arrow. Is it going to move to the right or to the left? So again, like I said, there's more solute on the right side. There's higher concentration of water on the left side. Therefore, the water is going to move wherever it's saltier. And the saltier side was on the right side. So the water is going to move to the right side. At the very end, you'll notice because the concentrations are not even, they're both equally as salty because the water moved across the membrane. The water moved to the right side to try to dilute out the saltiness on the right side. So if you have a YouTube, it's a YouTube. It's actually called a YouTube because of the shape of it. If you have a YouTube like this, you can actually separate the middle with a selectively permeable membrane and watch the water rise on one side because it's just saltier on one side. It's pretty cool. So there are three types of solutions. There's hypotonic, hypertonic, and isotonic. And as you can see, when you have these three types of solutions, different things happen when you put a cell into them. And when we talk about the solution, we're talking about the solution surrounding a cell. So if you pour a solution into a beaker and you put a cell into it, we're talking about that liquid around the cell. We'll probably never really talk about the actual inside contents of the cell, referring using these terms, but the solution of the outside environment around the cell is going to be described by hypotonic, hypertonic, and isotonic. So hypotonic solutions have a low solute concentration outside of the cell and a high solute concentration inside the cell. So that means the inside of the cell is going to be saltier. Okay, just leave it like that. That means the outside liquid that you put the cell into, that liquid inside that beaker is really, really not salty or really, really not sugary. It's kind of watery. Okay. If it's saltier inside the cell, the water is going to go towards that salt. So it's going to enter the cell. So water is going to flow into the cell. So we always draw the arrows to show that. So the net movement of water is into the cell. And the cell is going to swell and swell and swell until it can't handle it anymore. And then it will burst. And for animal cells, we don't call them bursting. We call it lysing, or it has already lysed. On the other hand, if you look at plant cells, because they have that cell wall, they're not actually going to burst. They'll just become really, really full. And then after they're really, really full, they'll just maintain their shape. That's when the plant is like standing really, really tall and nice and firm because these cells are turgid. So that's a hypotonic solution, when the outside has a low concentration of salt. So the inside of the cell is super, super salty compared to the outside. So the water is going to go in. On the opposite side, we have a hypertonic solution. So a hypertonic solution, there's a typo here. This should be hypertonic. Hypertonic solutions have a high solute concentration outside of the cell and a low solute concentration inside the cell. So a hypertonic solution means that the outside is so salty. So when you put that cell into that liquid is really, really salty. So the water is actually going to leave the cell. So the net arrow right here is actually an arrow leaving the cell. And if water leaves the cell is going to get kind of wrinkly and shriveled if it's an animal cell. Okay. If it's a plant cell, it has that cell wall, so it won't shrivel as much as an animal cell because it's stuck to the cell wall at certain points. We call that plasmolized. And that's when you have a plant that is wilted and sad looking because the water is actually leaving the plant cells and going outside the plant cells to that solution. Okay, because the outside solution is just saltier. So the water has to go out to try to dilute it and make it less salty to balance the inside and the outside, right? So the inside of the cell and the outside of the cell now have the same concentration, but the cell has been shriveled. So like I said, the net water flow is out of the cell and the cell is going to shrivel or shrink. The last or third kind of solution is going to be an isotonic solution. Oh, hold on. Sorry. An interesting way to remember a hypertonic solution is when you're hyper, you have a lot of energy. So when the solution is hypertonic, it has a lot of salt or has a lot of sugar. An isotonic solution means, well, iso means the same. If you ever looked at an isosceles triangle, it's a triangle that has two sides that are the same height or the same length. And iso basically means that the inside solution and the outside solution have the same concentration. That means the net water movement is going to be 0, but it doesn't mean water is not moving. It just means that water is moving in and out of the cell at the same amount. So if 2 come in, 2 waters come in, 2 waters are going to go out. If 3 waters come in, 3 waters are going to go out. It doesn't have to be the same waters. It could be any waters that are just floating around the area. But it means that if you were to cancel out the arrows, they would all cancel out, and then there would be 0 arrows left after you X them all out. Right? As you can see, there's 1 in and 1 out here. If I were to cancel them out, 1 in for 1 out, we would end up with no zeros after I cross them out. So there is no net water flow. Okay. So the cells actually can maintain their current state and they look normal and they don't change shape or they don't shrivel, they don't swell, and that's just their normal state. So what does it mean for cells? Like I've been saying, it's different for plants and animals because plant cells have a cell wall. So in an isotonic solution, the cells will stay normal in an animal cell, and in a plant cell, they'll be flaccid or just relaxed. When you put the animal cell into a hypotonic solution, the water is going to go in and swell and swell and swell and swell until it bursts because the outside is less salty, right? So the cell is salty, so the water is going to go in. But when the water goes into a plant cell, there's that cell wall to protect it from bursting, so it'll just become turgid or very, very firm and full. And when you have a hypertonic solution, because the outside of the cell is saltier, the water is going to leave the cell to go out. The cell is actually going to lose water and lose its hold on its shape. For an animal cell, it's going to shrivel and wrinkle. On the other hand, the plant cell will also shrivel, but it's not going to shrivel as much because it has that cell wall to kind of protect the integrity of its structure a little bit. It's still going to become a little bit soft though, and that's when plants or fruits or any kind of plant matter will wilt and become a little squishy. So here is the egg in vinegar.
Placed in a hypotonic solution versus a hypertonic solution. A hypertonic solution is simply a solution that has a lower concentration of solutes, dissolved substances, compared to the inside environment of a cell. For example, water is considered a hypotonic solution since it is very low in solutes. A hypertonic solution is a solution that has a higher concentration of solutes compared to the cell's inside environment. For example, water with a lot of salts is considered a hypertonic solution since it is very high in solutes. How can we examine the effects of a hypertonic solution on a cell? You might be thinking, well, I'm made out of 50 to 70 trillion cells. If I drink this, I'll know what happens to cells in a hypertonic solution. But do not do that. Drinking salt water can be very dangerous as it severely dehydrates you. Nausea, vomiting, and even hallucinations are just a few symptoms of drinking massive amounts of salt water. Ask anyone who's been stranded at sea, which is unlikely because people who drink seawater eventually died due to kidney failure. So what you really need is a lab animal to conduct your test. Darn, she's too smart. Time to try her older brother. Well, that was a bad experiment. What's a better way to test how a cell reacts when placed in a hypertonic solution? Some eggs, like chicken eggs, are surrounded by a hard outer membrane which protects the semi-permeable inner membrane. This membrane is semi-permeable because it allows substances to leave and some substances to enter. In particular, the semi-permeable membrane allows water to both enter and leave the cell, but large molecules, like salt or sugar, are too big to pass through. The question is, how can we examine the semi-permeable qualities of this inner membrane with the hard eggshell in the way? Dissolve the outer shell by placing an egg in vinegar. Vinegar has a chemical called acetic acid. Eggshells contain calcium carbonate. When calcium carbonate in the eggshell comes in contact with acetic acid, a chemical reaction occurs. Calcium carbonate will break down to form carbon dioxide, water, and calcium acetate, thus dissolving the calcium carbonate shell. Within seconds, you'll see bubbles form. These bubbles are carbon dioxide that are produced when acetic acid breaks down the eggshell. Within 3 days, you should have a semi-permeable membrane free of the hard outer shell to conduct your experiment. Before starting our experiment, we will first mass our egg using a scale. Egg A has a mass of 94 grams. Egg B has a mass of 103 grams. Egg A we will place in a hypotonic solution, water. Egg B will place in a hypertonic solution, thick and sugary corn syrup. Egg A now has a mass of 97 grams. Egg A increased slightly to 103% of its original weight. We can conclude when a semi-permeable cell is in a hypotonic solution, water will enter the cell. Egg B now has a mass of 59 grams. Egg B decreased to 57% of its original weight. We can definitely conclude that when a cell is in a hypertonic solution, water will leave the cell. In fact, this is evident by the layer of water sitting on top of the corn syrup. However, 103% change in mass is not very significant. So how about we try this experiment again, but switch the eggs? This time, egg A is going in the hypertonic solution, which is still on the right. Egg B is going in the hypotonic solution, which is still on the left. After several hours, mash your eggs again. Egg A went from 97 grams to 55 grams. Egg A has decreased to 57% of its original weight. Egg B went from 59 grams to a whopping 105 grams. Egg B has increased in weight to 177%. From this experiment, we can definitely conclude that when a cell is in a hypotonic solution, like water, water enters the cell. On the other hand, when a cell is in a hypertonic solution, like corn syrup, or more realistically salt water, water will leave the cell. Now that you're done with this experiment, time to play with those shellless eggs. No!
Okay, so that was a demo that was showing what happens when you place a cell, for example, an egg, which is one cell, even though it's kind of hard to believe, into a hypotonic and a hypertonic. And when you put in hypertonic, the cell will lose water and it'll become all soft and wrinkly. And if it's in a hypotonic solution, water is going to go into the cell and the cell will become as big and as full as possible. And if water keeps going in, then it's going to burst. So we're going to do a let's try. So I'm going to ask that you label the solution as hypertonic, hypotonic, or isotonic. You're going to draw an arrow to indicate the net movement of water, if it's going in, out, or neither. And you're going to explain if the cell is going to swell, if it's going to shrivel, or if it's going to remain the same. So let me do this first example. So is the solution in the beaker hypertonic, hypotonic, or isotonic? Well, to answer that question, we're going to look at the solution on the right side. So the outside is 3% salt and the inside of the cell is 1% salt. So we're looking at this solution on the outside right now. Is it saltier, less salty, or equally salty? Okay. After that, we're going to draw the arrow that shows where water is going to be moving. Water is going to move from the less salty to the more salty area. Okay, it's always going to move from the less salty to the more salty area. And then based on the arrow, we're going to describe if the cell is going to swell, shrivel, or remain the same. So we're labeling the outside as hypertonic because 3% is bigger than 1%. So it's saltier. And if the outside solution is saltier, remember, it's going to be hypertonic because it's got more salt. It's got a lot of salt in it, like having a lot of energy when you're hyper. Now, because the outside is saltier, the water is going to move from the less salty to the more salty area. So it's going to move from inside the cell to the outside of the cell. Remember, your arrow has to cross the membrane. Then based on the arrow, well, water is leaving the cell. So the cell will shrivel and potentially crenate. Right? If it wrinkles enough, it's going to crenate. It's going to become a little raisin of a cell because the water is going to leave it until the concentrations are equalized. Now you are going to try. You're going to take this example. You're going to label the solution again as hypertonic, hypotonic, or isotonic. You're going to draw an arrow to indicate the net movement of water. And you're going to explain if and why the cell will shrivel or swell or remain the same. Now, again, as a hint, you're going to think of, is the solution in the beaker hypertonic, saltier, hypotonic, less salty, or isotonic, the same saltiness compared to the cell? So you're going to look at the numbers here for 7% salt in the cell and 5% salt outside of the cell in the solution. So we're talking about this blue solution on the outside. Okay, so you're going to pause and go ahead and do that inside of your Pear Deck. I will be checking these answers. Okay, and the next question here, again, you're going to be doing the same 3 steps. Label the solution as hyper, hypo, or isotonic. Draw an arrow for the net movement of water. Is it going in, out, or is there zero movement? You can draw in and out if you need to. And explain if and why the cell will swell, shrivel, or remain the same. So the hint is still the same. We're looking at a 12% salt cell instead of a 12% salt solution. So you're going to be looking at this blue solution on the outside. Is it going to be hyper, hypo, or iso? You're going to pause and do that, and I will be checking this for completion and correction. The last one that you're going to work on is going to be this scenario. So there's a 1% salt in the side of the cell, and the solution that we're focusing on is a 10% salt solution. So you're going to tell me, is that hyper, hypo, or isotonic? You're going to draw an arrow to show me if the water is going to move into the cell or the water is going to move out of the cell, or it's going to move back and forth, so technically 0. And then based on that, you're going to tell me, is the cell going to swell, shrivel, or remain the same? So you can pause and go ahead and do that. But this is the last part of the slide set. I will be checking that you completed all 3 of these practice problems at the end, and if you did any of the stuff in the beginning too. That's for completion. And then you're going to go ahead and move on to your interactive notebook section, which would be section 2, which covers osmosis. And you will go ahead and do your edge ticket at the very, very